So seminar tomorrow, noon. Kiki Sanford's how she got into science communication. Next week is Eugene Koonin, both at noon in the biology seminar series and in the evening down at the First Congregational Church about how viruses are critical for life as we know it. You can get your tickets starting tomorrow for that over at the box office. Second thing is that it's not just the biology department that has cool seminars. The seminar speaker in chemistry this week is Professor Talbot from UC Riverside. He's going to be talking about stem cells and particularly interactions of small molecules with stem cells and looking at toxicology and those kinds of things. So lots of cool stuff going on. This is Friday at 3:15, so after our next lecture, so I'll remind you about that. What's happening a week from Friday? Exam, exactly. So next Wednesday will be review, and the material to be on the exam will be up through next Monday. So other questions about that before we move on through? Exam, same kind of format, 50 questions, multiple choice, horrendous questions. Okay, good. So to give you some practice on the kinds of horrendous questions that there'll be, our clicker questions for today are, which of the following does not have a homologue in archaea? TBP, TF2B, RNA polymerase II, TF2H, or Stedman's favorite, A and B. Oh, sorry, I didn't give you the countdown. My bad. Got to do it again? Oh, yeah, clearly. Have to do it again. No question about it. Yeah, everybody misses out on that when they listen to the recording, of course. So what do people think here? In fact, they agree with me for the most part, which is nice. So it is TF2H. Why is it TF2H? Two reasons. One, the archaeal polymerase does not need a helicase, which is one of the things that TF2H does. What else does TF2H do? It has a kinase activity which phosphorylates what? The C-terminal domain, CTD, of RNA polymerase II. Archaeal RNA polymerase does not have a CTD, so you don't have that. TF2B, there seems to be a little confusion about that. TFB binds to the BRE, which is present both in archaea and eukarya. So, next clicker question, and I will count this one down, I promise, is to identify the 3' end of the exon to be joined to the following exon. It is bound by U1 snRNP, U2, U3, U4, U5. 1 minute. Thirty seconds. $15. 10. 5. Okay, so see, I can see the results up here, so I have a bit of an idea how things are going. One advantage of actually being here for class is that you're actually able to see this as opposed to anybody who's just going back and listening to this online, because I don't have that here. What I would do in a case like this is to turn the light on, draw out a exon, an intron, and an exon. So this is my getting away from having a whiteboard. So the key here is thinking about where's the 5' end of your exon, where's the 3' end of your exon, 5' end of the intron, 3' end of the intron. So we think about what's going on in the intron. We've got the branch point, and that's going to bind to what? The branch binding protein and U2AF, but most importantly for this question, what? U2, SNRNP. So that's binding here. What binds here? U1 is binding here. And so that is the 3' end of the exon or the 5' end of your intron. It's always going to depend on how you're thinking about it. Does that make sense? People seem to... So it is U1 because the 3' of this exon. Okay, so let's switch back over. Boom. HTMI. So now all of you here are going to do so much better on that one question on the midterm. But I'm not sure. So again, the confusion seems to be between U1 and U2, and that's why I wanted to draw it out on here. So the correct answer is U1, not U2, because U2 is binding at that branch point. And it turns out also very close to the end of the intron, so the 3' end of your intron. Okay, so now all of you can leave. Yes?
So looking at how the diagram is then laid out,
Mm-hmm.
it said 3' splice site. That was then for the intron itself then?
So if people talk about splice sites, and I will actually switch back over here, again, to give you great advantages. When people talk about splice sites, they're going to talk about this and here, but that's going to be relative to your intron, not relative to exons.
Thank you.
Okay, so just really briefly, I thought this was totally cool and fit in really nicely to the lectures. This is, in fact, came out in an early edition in Nature on February 1st, so we're pretty close to being up to date. This is the reconstruction of U4, U5, and U6, the snRNAs, but also all of the proteins that go with them. So this is a RNP, ribonucleoprotein complex. One of the things that should hopefully be really obvious here is it's not just the RNAs. In fact, the RNAs here, which are in blue, are relatively small relative to all the proteins that are in here as well. This is that complex that comes in after you have U1 and U2 and basically does the job of moving all of the different snRNAs around, which are base pairing to your intron and exon, and then also catalyzing the reactions of first joining up to that branch point, and then the lariat getting removed and rejoining the exons. So this is really the business end of the spliceosome. We're still trying to put everything else together to figure out exactly what the whole structure is. That's not completely known yet. So it's a very active area of research, and it reminded me that basically, well, not quite every slide that I talk about here in class is something we could spend a whole term talking about. Certainly splicing is something we could easily spend a whole term talking about. So this is an introduction to molecular biology. We can't, by any stretch of the imagination, do everything. So last time we talked about splicing and capping. Talk a little bit more about splicing today, but mostly today we'll talk about polyadenylation, RNA export, and a little bit of RNA, ribosomal RNA modifications, which also has to do with RNA-RNA interactions and RNPs. But first wanted to talk about where introns and splicing probably came from originally, and thinking about everything from an evolution point of view, which is the reminder that this is the week of Darwin Day. Darwin Day itself is on Friday. There will also be an evening seminar by our excuse me, biology seminar speaker talking about how evolution is absolutely critical to everything we know about. And why is that? That actually has to do with some of the studies that were done. A guy by the name of Tom Check, when of course you say who did the experiments, it wasn't really him, it was all of his graduate students and postdocs who did this. They were just looking at RNAs and trying to isolate some of these spliceosomal components. And what they found was, and apparently the graduate student went and told the big guy who ended up getting a Nobel Prize for some of this work, that the RNA that they had was splicing itself. And the boss said, no, that can't be true, go back and do it again. And go back and do it again, go back and do it again, go back and do it again. And eventually they figured out that there were specific RNAs that could literally chop themselves out of another RNA and ligate these ends back together. And so it was just purely RNA. So that last slide that I showed you with all the proteins hanging off of the RNA, you don't need all of those proteins. They stimulate the reaction, but they're not necessary. And this was some of the very first evidence that RNA itself could catalyze chemical reactions. And so this process was what was first known as one of the first ribozymes. Yeah?
So is the protein complexes not needed for any of it? Or are there specific ones that it's not needed for, like particular sequences that can self-splice, but not all RNAs can self-splice?
So the question here is basically, can you get rid of all proteins for all splicing? That's just paraphrase your question. Yeah, and the answer is no. So probably what has happened, again, through evolution, is that proteins have been recruited to help with splicing. And in that process, you don't need all of the other RNA sequences. So you just need a very few specific RNA sequences. Probably the sequences are really critical for catalysis. And stimulating that catalysis are then have been lost. But what has happened is you've generated a much more general system. As we talked about last time, the splicing happens all over the place now. And that's because the consensus sequences are relatively short. And you can have splicing that happens all over the place. The big problem is controlling it. Rather than here, it's a very specific sequence, which is going to fold up into a very specific structure and give you that very specific function. And so these are what are called self-splicing introns I have up here at the top. And so they can splice just by themselves. You make the RNA, purify the RNA, it will splice itself. Now, for most messenger RNAs, that's not the case. You need then the spliceosome. You need extra RNAs to come and help do that. Yeah?
Is the splicing a product of the environment it's in, or a sequence that the RNA has?
So the question here is, is there some, basically, again, I'll paraphrase your question, is there some signal that leads to the splicing, or is it just something which is intrinsic to the RNA? And the answer here for these self-splicing introns is intrinsic to the RNA. So if you have an RNA that's, well, the appropriate sequence, it will splice itself out. Now, you can play around with temperature and, actual concentrations of different ions, but it will happen. Okay, so this was the first one that was found. Turns out that instead of having that nice little bulged A in it, you actually have to put in an extra G, and so that's actually thought of, how you can get some regulation here, extra GTP. But then later people found these other kinds of self-splicing introns, which are called group 2, because this is one first kind they found, the second kind they found. These guys work almost identically to the way the spliceosome works in terms of an A that gets bulged out, in terms of the 2', 5' bond, etc. And so this is probably what then evolved into our spliceosomal machinery that we have right now. So that's it for splicing. I wanted to talk now about what happens at the 3' end of your messenger RNA. This is our poly-A tail. We've talked about before. How are poly-A tails made? Basically, poly-A tails are made just like splicing and capping as the RNA is being made by the RNA polymerase. It's a co-transcriptional process, and basically the RNA polymerase will transcribe this sequence, which has a consensus sequence over here, consensus sequence over here, and then a specific spot that you have an endonuclease activity that will cut the messenger RNA as it's being made. Once that gets cut, you have a 3' OH. There's some specific proteins that we'll talk about in just a second that add a whole bunch of adenines to it, and then this piece gets chewed up. And so unlike transcriptional termination in bacteria, where you've got a nice structure in your RNA, the hairpin loop and the U's, which will sort of pull the RNA out just by forming that secondary structure, what happens here is there are exonucleases, so they chew in at ends. So we mentioned before, it's one of the reasons you have a cap, is to block all of these exonucleases, that chew up from 5' to 3'. And so basically they're chasing the RNA polymerase. The RNA polymerase is over here making more RNA, and the exonuclease is chewing in at this end. And it turns out it chews in to a certain point, and then the RNA falls out of the RNA polymerase, and the RNA polymerase terminates and goes back, tries to find another promoter, etc. And so very different mechanism of termination, and termination and poly-A tailing are clearly very closely linked to each other. If you don't have an endonuclease that cuts here, you're never going to terminate because you don't have this exonuclease which is chewing in at the ends. So let's look at how that happens. First, you have these conserved sequences in the RNA as it's being made that some more acronyms bind to. The cleavage and polyadenylation specificity factor. This is what binds to that sequence which is in the messenger RNA that's going to be staying in the messenger RNA. You'll notice I don't mention the sequence per se, so I don't think you need to know it. However, there are a couple of things on red on here. I would know what these are. So the cleavage and polyadenylation specificity factor. This basically says here is where we are going to make that endonucleolytic cut. There's the second conserved sequence, which binds to CSTF, which stimulates this cleavage effect. And then once you have cleavage that takes place, poly-A gets put onto that 3' OH. That's done by a poly-A polymerase, so just puts A residues on. This is a non-template specific RNA polymerase because it doesn't have a template. It just puts on A's and eventually will fall off. And then we have a poly-A binding protein that does exactly what its name is, binds to poly-A tails. That would be the PABP, and sorry about people over here, it's the red thing down at the bottom, poly-A binding protein. So that's the process. How do you make sure, again, totally anthropomorphizing here, that this happens in the right place? As I mentioned, it's all happening co-transcriptionally. So as the RNA is being made by the RNA polymerase, so you have your CTD phosphorylated by TF2H, which binds to all of these proteins that you need to modify the RNA, including CPSF and CSTF. They're bound to the CTD, and it turns out the way they get to the CTD is they interact with TF2D. So when you're at a promoter, some of the first things that bind are going to be TF2D together with the TATA binding protein that also will have these extra proteins that you need to put onto the RNA polymerase. So when it's cruising along, it can do poly A tails. Turns out also the same thing is true for splicing and for the capping enzymes. So where are all these things? Again, they're associated with the phosphorylated C-terminal domain of the RNA polymerase, phosphorylated by TF2H. One thing is getting the polymerase away from the promoter. You're just phosphorylating. Phosphates, nice negative charge. DNA, nice negative charge, is repelling each other, will help the polymerase to move away. But probably at least as important is binding to all of these other factors, and factor means protein, which are involved in messenger RNA processing. So turns out the capping enzymes, the guanylyl transferase, the phosphatase are associated with the CTD. Splicing proteins are associated with the CTD. And as we just talked about, the tailing proteins are all associated with the CTD. A lot of people talk about this as being a messenger RNA factory. So it's not just the RNA polymerase, but all of the proteins that are involved in taking that primary transcript that's got no cap, no tail, all of the introns in it, and making it into something which can eventually be translated. And some of the data for this are that people have really nice, both in vitro and in vivo. When I say in vitro, that means basically in a test tube. In vivo is in a cell. That some of these factors are going to interact directly with each other. So it turns out that this tailing factor also interacts with some of the factors which are important for splicing. So all of these guys are all together bound to this CTD. And I like this image in particular because it shows that the CTD is really a pretty huge thing. And we looked at it at the very beginning of lecture last time. It's a long stretch of amino acids that lots of other proteins are going to be binding to. Another thing that people have seen is if you just look at cells, we'll look over all the way over here on the right, and try and localize where all of these modification proteins are. So your snRNPs, your tailing proteins, your capping proteins. Turns out they form little dots in the nucleus. And those dots are probably where you have not just one RNA polymerase together with all of its modification machinery. There's probably a whole bunch of RNA polymerases that are associated with each other, probably through some of these scaffolding or aggregation proteins. So each dot that you see over here is going to represent one of these things, and also emphasizes that the DNA is really moving, whereas the polymerase complexes, the modification complexes, probably aren't moving very much. And again, it makes sense if you think about size of the proteins relative to the size of the nucleic acid. It makes a lot more sense to move the small thing instead of trying to move the big thing. Okay, so now we've made our messenger RNA. It's in the nucleus. This is a micrograph of the nucleus. We need to get it out. How does it get out? Well, through RNA export. So a couple of things that have to happen in this process. The first one is binding by, yet another acronym, the cap binding complex. So we made the cap through phosphatase, the guanylyl transferase, the methylases. Why do you make the cap? It's to protect it, but also to help it get out of the nucleus. And that binds to the cap binding complex, which is one of the most critical things in terms of getting this RNA out of the nucleus. There are a couple of things about getting RNAs out of the nucleus which are really important. You want to make sure that it's the right RNA, it's been spliced, etc. Because if you don't do splicing and you take that RNA out into the cytoplasm, it's going to get translated. And you don't want to translate usually most of the introns, etc. So you want to make sure that all of the splicing has already taken place. One way, and we've already talked about this already, is that SR proteins. What do SR proteins usually associate with? SR proteins associate with exons. So SR proteins associate with exons. So if your RNA is bound to SR proteins, it's likely to have exons in it. So it's likely to be the right kind of RNA. Of course, it still binds to exons and introns as well. You don't want to have a bunch of introns. And one of the ways that you can tell that you have introns is, we talked about last time, you've got these HNRNPs that bind to introns and bind specifically to introns. So if you have HNRNPs associated with your RNA, most of those are going to say, hey, this is not the right thing to export. Of course, the problem with HNRNPs is that they bind everywhere because they're heterogeneous. There are all kinds of different ones. So some of them are bound, some of them are not. But more important here, for our purposes, SR proteins, poly-A tails. How do you know you have a poly-A tail? Because you've got the poly-A binding protein that's associated with it. And then this last thing here, again, far too many acronyms, EJC, it's an exon junction complex. So once you've gone through the whole splicing process, we put two exons together. Now you have binding by another set of proteins that basically says, hey, a splice has happened here. This is good. And we're ready to export this whole thing from the nucleus. How does that happen? Nuclear transporters that you've already learned about in cell biology, or learn a lot more about in cell biology next term, which will then lead to transport of this whole complex. So it's not just the messenger RNA, it's the messenger RNA plus the cap binding complex plus the poly-A binding protein plus also these exon junction complexes, which all be exported from the nucleus through the nuclear pore. And this is just a cartoon of what they show when people have looked at these things. But the basic message here is it's a pretty darn big thing. It's not just the RNA. It's RNA plus all of these proteins. So what we have now is this nicely exported RNA. This is now ready for translation, which is the last part of today's lecture. But what about all of the introns and that end piece that's still stuck to the polymerase after you've cut it with your endonuclease to make poly-A tails? Turns out you've got a lot of extra RNA floating around in the nucleus, which is not a very useful thing to have. So you have the RNA garbage disposal unit, which is also called the exosome. Again, people love these zomes. Molecular biologists love zomes. Basically what this is a whole collection of RNA exonucleases. And as we mentioned, exonucleases chew in from the ends of nucleic acid. And in this case, it's both 5' to 3' and 3' to 5'. Anytime you have a non-protected end, particularly of RNA, inside the nucleus, it's going to get chopped up by this exosome complex. Okay, that's it for messenger RNAs. Now we're going to move and talk a little bit about ribosomal RNAs. Yeah?
What does it spit out when it chops up the RNA?
Oh, so the question is what does the exosome spit out when it chops up the RNA? They're now going to be monophosphates. So AMP, CTP, UTP, TTP, no, TTP, keep saying that, CTP. But the four nucleotides as monophosphates, those then need to get turned into triphosphates, and then they can be used again for transcription purposes. Okay, so let's talk a little bit about the ribosomal RNA. Ribosomal RNA is, we mentioned before, hopefully you're still remembering, good review for the exam, the most common RNA that you have in any cell is the ribosomal RNA. And that's because it's, of course, involved in translation. As we'll see probably on Friday, basically it's the RNA in the ribosome, which really does the job of making the proteins. Because you have so much of this RNA, and you don't have the advantage of amplifying it through translation, you need many, many copies of this ribosomal RNA gene. In our case, we have 200 of them. E. coli has 7. So just transcribing them like mad is not good enough. You have to have many copies of the genes. So we talked about before, RNA polymerase 1 in eukaryotic cells is a very specific polymerase that makes most of the ribosomal RNA genes. Turns out there's one which is not being made by it. It's made by RNA polymerase 3. But the vast majority of the ribosomal RNA in eukaryotic cells is made by RNA polymerase 1. RNA polymerase 1 makes this one big precursor ribosomal RNA. So this should sound kind of familiar. Lots of messenger RNAs are made as these precursors as well. There are quite a few pieces here which are actually not used in the final version. But instead of what happens with messenger RNAs, here, these guys just get chopped out. They don't get religated back together. So it's just an endonuclease cleavage that happens. So that's the first thing that happens. But then, and this is why we're talking more about it, this ribosomal RNA gets heavily modified. And that's what all of these triangles and dots here are supposed to represent. Once you have all of these modifications, now you can get the 4 ribosomal RNAs that come together to make the ribosome. Again, 5S is made by RNA polymerase 3. All these modifications that happen here, they happen through the activity of more RNPs, just like the case with splicing, only these are now these small nucleolar. So this O here is the big difference. SN RNAs, those are the ones that are important for splicing. SNO RNAs are the ones that are important for ribosomal RNA modification. So let's take a look at these. Again, there's SNO RNAs together with proteins make SNO RNPs, so ribonucleoprotein complexes. That's supposed to be shown down here. The RNA is what's in blue, and the protein is the blob that goes with it. There are lots and lots of these. We're only going to talk about 2 particular kinds here. These are the so-called hairpin, which is what you see in the middle here, ACA SNO RNAs, and the box CD RNAs, SNO RNAs. Can talk about them, why they've got these names later. It's not important for this class. What these guys do, the RNA and the protein, will do 2 of the most common modifications that you have in your ribosomal RNA. One is the formation of pseudouridine. So normally, uracil is going to bind to your sugar over here. And basically, what this protein does is takes the uracil, flips it over, and attaches this carbon here. So this is your pseudouridine. This was one of the most common, about the same level. A couple of hundred you have of these in your ribosomal RNA. About a couple of hundred you also have O-methylribose. O-methylribose should sound really familiar. Why? Where else have we seen O-methylribose? In cap structures. So caps have O-methylribose. So turns out that methyl is put on by a completely different mechanism. So why bother with all of these RNAs? Why do you want to have RNAs? What's the great thing about RNAs? DNAs too. What do? They base pair. And they base pair specifically. So it turns out there are hundreds of very specific places in the ribosomal RNA where you need a pseudouridine or you need a 2' O-methyl ribose. How are those found and modified? They're found and modified because you have base pairing interactions between the SNO RNA and the sequence which you're going to modify. Because we've got hundreds of different places in the ribosomal RNA that need to be modified, you have to have hundreds of different SNO RNAs. So for each of those places you're going to modify, you're going to have a complementary sequence in your SNO RNA to the ribosomal RNA. And that's going to bring the protein, which will give you your pseudouracilation or your O-methylation. So that's the modifications that happen to your ribosomal RNAs. Where does all this happen? Why do you call them these snow RNAs? Because it's in the nucleolus, which is basically where ribosomes get made. So you have all of the RNA polymerase 1 in eukaryotes is in this. So nucleolus is supposed to be the gray thing here in the middle. You have transcription of all of these ribosomal RNA genes. Those get processed with these snow RNAs. And it turns out that the ribosome themselves get assembled here too. So it's not just the RNAs that come together, but the proteins as well. And these then end up getting exported through the nuclear pore. How this happens is really an unknown process, because when we look at the ribosome, it's actually pretty darn big, and the nuclear pore is actually rather small. So how do you get a whole ribosome subunit out through the nuclear pore? And the answer is we really don't have a good handle on that. Yeah?
So the entire ribosome and all the rRNAs that are going to work with it all come out at once?
Yeah, so the question is here, basically what is coming out there? So it's the ribosomal RNAs, which have all been modified, that then get put together, and then all the proteins get put on that. And so to get all the proteins there, all the protein synthesis is happening out in the cytoplasm. So you've got to make the messenger RNA for the ribosomal protein, splice it, send it out to the cytoplasm, have that get translated, get that protein imported back into the nucleus, assembled with the ribosome, and then the whole thing sent back out again. It's a really baroque process, but it works. Like all of evolution, it just has to work. It doesn't have to be the best. It just works. Okay, so that's messenger RNA processing. Any questions, concerns, comments, worries? Yes, it will be on the exam.
The large subunit that gets exported.
The large and small subunit of the ribosome both get exported.
And are those the smallest subunits? Are there separate domains of those? Or
Okay, we'll talk much more about the ribosome. Sorry to obfuscate here for your question, but we'll talk a lot more about the ribosome and all the different subunits coming together later on. But basically, there are two subunits of the ribosome. There's a large subunit and a small subunit. And those both get exported separately, and then they come together, as we'll see, for translation. Okay, so, meine sehr geehrten Damen und Herren, Mesdames et Messieurs, let's talk translation. Translation's all about going from nucleic acid language to amino acid language. And doing that, you need your dictionary or Google Translate in order to get from that one language to the other language. What cells use, of course, is the genetic code, and we'll talk about the genetic code. No, you don't need to memorize the genetic code, not in this class. Yay, something you don't need to memorize. We'll talk a little bit about ORFs. ORFs are open reading frames. A lot of people confuse these with genes. This just has to do with how you interpret your genetic code. How that happens, of course, is the tRNAs, the little adapter proteins, my little decoder ring. Those people who know Cracker Jack, that's what you find in the Cracker Jack box. How are those tRNAs translated? It's all about tRNA synthetases. And then we'll talk about the ribosome, which actually does the job, and then talk a little bit about the process. The translation cycle, someone was asking me about this in office hours the other day, is actually really similar in the very overall big picture to everything else we've talked about so far. You need initiation, you need elongation, you need termination. You need that for replication. You need initiation, elongation, termination. For transcription, you need initiation, elongation, termination. Now, they're different processes, but it's all because you've got to start somewhere. Once you've started, you need to move along and then get to the end. And that's true for all of these molecular biology processes that we've talked about. So we'll talk about initiation. That's how you get the first amino acid. and that's where you get the two subunits that come together. Elongation, which is polymerizing your amino acid chain, and then once you get to the end. And so we'll talk about all of these as we move through here. But first, we need to talk about the genetic code. How do you go from the nucleic acid language to the amino acid language? And we've only got 4 nucleotides, as we decided way back when, before the first midterms. We've forgotten it all already. We only have 4 different nucleotides, and they're kind of dull and boring, and so they probably aren't the genetic material, till people showed that it was. And then 20 amino acids, which are much more diverse and have all of these different possibilities. And people knew about this very soon after looking at the genetic code and what we had in terms of DNA, what we had in terms of protein. So people tried to figure this out. A lot of the early molecular biologists were physicists. So they said, you know, physics, we've solved everything, we've made an atom bomb, let's move to something more interesting, biology. And a lot of them are still doing it. So basically, they just did some calculations. If you have 4 nucleotides and only 1 nucleotide encodes 1 amino acid, you can only have 4 amino acids, right? Because, you know, C's are going to give you 1 amino acid, etc. If you've got 2, that would give you 16, but that still doesn't give you 20 amino acids. So you have to have 3. But 3 is way too many. 64 possibilities of 3 nucleotides. So what's going on with that? And a lot of people spend a lot of time thinking about how all of these things come together. And actually probably only makes sense in terms of evolution and the evolution of the genetic code. And if you're interested in more details on that, take Dr. Lehman's class in chemistry about origins of life and origins of the genetic code. So, so it was clear that you needed to have 3 nucleotides, and those 3 nucleotides were going to make up the genetic codes. You need 3 nucleotides per amino acid. The next question that came up was whether you had an overlapping or a non-overlapping code. And what that means is, if you have sequence here, AGC, ATCG, do you use nucleotides 1, 2, 3, and then 2, 3, 4, and 3, 4, 5 as giving you individual proteins? And originally, that made a lot of sense because, as we've talked about before, I should have brought my DNA model. DNA is pretty inefficient. If you need 3 nucleotides for every amino acid, and it's this really long thing, it actually makes sense to use one of these overlapping codes, because from this original sequence, just 7 nucleotides, you can get 5 different amino acids out of it. If you don't have an overlapping code, you just use 1, 2, 3, and 4, 5, 6, you end up with a lot fewer amino acids, and it's a lot less efficient. Turns out, of course, that this is what we use, and probably the reason for that is it's a mutation prevention mechanism. Because if you have 1 nucleotide change, again, it's hard to see down at the bottom here, you change 1 nucleotide here, you could change 3 different amino acids if you use an overlapping code. Whereas with a non-overlapping code, you only have 1 amino acid that changes. And it turns out it's probably that mutation avoidance, and I shouldn't really say mutation avoidance because the mutation is just a heritable change you have in the DNA. But in terms of changes that you get in proteins, those are much, much fewer if you have a non-overlapping code. The other thing, of course, with the genetic code is that, again, we've got way more codons than we actually need. And when I say codon, that's just the sequence of nucleotides from 5' to 3' that is going to say, hey, this should be a particular amino acid. A couple things to mention here. Again, the big circles are probably things to concentrate on. I wouldn't be worried about the sequence here. But what you can see if you just look at the genetic code, most amino acids just have 2 different codes that code for them, but there are some exceptions as well. Probably the most important one, and one that I would remember, is this one here, AUG for methionine. That's because that's where proteins start. And so that is one of the 2 where we have just 1 stretch of 3 nucleotides is going to give you one specific amino acid. Turns out that tryptophan is one of the least common amino acids, and that's probably why it also only has one in most genetic codes. Another thing that you have to have here, because we already talked about initiation, elongation, termination, are certain codons, which people also will call nonsense codons, because they don't code for an amino acid. So these 3 here, and it's not important what their sequences are, basically say stop. This is it. Again, there are lots of different ways of looking at this code. Oh, again, the other thing was that some amino acids have a whole bunch of codons. Arginine has 6, and a few other ones have 6 as well. Yeah?
Excuse me. It seems like a good math problem to compute like the least overlapping possible code that could be done with the 4 letters and compare with the natural system of the code.
Okay, so the question here is about there should be relatively straightforward mathematics to do the calculations and figure out how you can get the most efficient code or what could actually be there. It turns out that's what some of the people very early on, in fact, did. Sidney Brenner is one of these people. He did some of those calculations and basically showed just through calculating that an overlapping code was impossible. So, in fact, there's a nice paper called The Impossibility of an Overlapping Code. If you're interested, I can post it. Yeah, you had a question?
Yeah, I was just curious of, like, what you meant by, like, impossible. So, like, these things don't actually occur in nature?
Yeah, and so what they did is he looked at all of the possibilities. Sorry, repeat the question. So how did he come up with being impossible? And so he looked at all the possibilities, and just the fact that you have 20 different amino acids, and you look at the number of different dinucleotides, dipeptides that you could have, what amino acids next to the other one, and the number of possibilities that you can get there versus the number of possibilities you could code with different overlapping versus non-overlapping. And it turns out with an overlapping code, you can't code for all of the possible 2 amino acids next to each other. And, okay, it's possible that that's not true. You don't have these 2 amino acids next to each other, but people then found that there were these 2 amino acids that were next to each other. Yeah, you had a question?
Yeah, is there any, conceptually, is there any significance to, like, tryptophan having very similar sequence to the stop codon? Like, you can do 2 single swaps and 1 double swap, and then your stop codon is that?
So that's actually a great lead-in to this way of looking at the genetic code, which I like a lot better. So basically the question is, what's the significance of tryptophan being very similar to the nucleotides that you have in stop codons? I'm going to refer you to Dr. Lehman about that, or we can talk about it that's offline here. There are some really interesting aspects about that, but we won't get into them here. So I prefer looking at the genetic code this way. And partly this also gets to that whole mutation avoidance process, because the code is degenerate, i.e. you've got lots of codons which can code for the same amino acid. One of the things that people noticed also really quite early on, you remember G plus C varies quite a bit, that's what Chargaff found, but it turns out the percentage of amino acids from bacteria to humans really doesn't vary very much. And so you actually have some organisms that are going to use a lot more of some particular codons, and other organisms use a lot of different codons, but you still end up with pretty much the same amino acids. We already mentioned 6 leucine codons, 1 tryptophan codon, 1 methionine codon, and 3 stop codons. But what about this mutation prevention business? And so the easiest way to look at that is here with, I think, this genetic code table. First letter of your code on this axis, second letter here, third letter here. So if you're just looking for one particular amino acid, say you start out with an A, that's your first one, gives you this set of amino acids. This is color-coded based on the chemical properties of each of those side chains. And one of the things that should become really obvious here is that the colors are all close together. All these brown guys, which are the hydrophobic amino acids, are really close to each other. These guys here, serine, tyrosine, threonine, asparagine, these are our hydrophilic, and also our friends down here, the charged ones. Remember? Aspartic acid, glutamic acid. arginines, lysines, they're all grouped together. And what that means is, so here, for instance, second codon U, that gets us down to here, and then of course the third G. Why did I pick this one? Because it's AUG for your methionine. But what this means is you can have changes, for instance here, if you have an AU to start with, you've got 3 different possibilities for what the third codon can be, but it's all still going to be isoleucine. And even if you do have changes, for instance, in the first nucleotide of your codon, it's going to give you something which is pretty similar in terms of its chemistry, which is going to give you a pretty similar structure and probably a pretty similar function. So it's a really elegant way, and again, the evolution of genetic code is a really fascinating process, way beyond the scope of this class, to make sure that your proteins stay pretty much the same even when you've got all of these changes that happen to your DNA and all the DNA damage, because we happen to live in an aqueous and relatively warm and oxidizing environment. Yeah, your question.
So when you say mutation correction, these mutations, are they coming mostly from the replication cycle or the transcription cycle, or is this actual environmental?
So it's whatever changes have happened to your nucleotides. Could be a mistake in replication, could be a DNA damage thing. There are all kinds of potential ways that you could have these changes, but you're not going to see very many of those changes in your proteins. So also means that evolution takes a while for some of this stuff to happen. Is there a question or a stretch over here? More of a stretch. Cool. So how do you decode the genetic code? It's our fancy little decoder ring. We have to go from this. So anybody read Greek? Good. Let's just translate my Greek letters. How do people figure this out? Basically, all they did was take RNA of a specific sequence, put it together with ribosomes, and see what sequence they got back out. And the first one of these was poly-U. So whole stretch of uridines. You put that into a setup which has ribosomes, all the amino acids. What do you get out? A whole bunch of phenylalanines. Oh, cool. That means UUU codes for phenylalanine. And you can do this again and again and again and eventually figure out the whole process of the genetic code. So that's what people did. Then they figured out that you have this fancy schmancy adapter, which has binding sites for the codon at one end and the specific amino acid at the other end. One thing to remember about this tRNA is that whenever you've got base pairing interactions, they're always going to be opposite to each other. And we'll take a look at that again in just a second. So that tells you what the code is. UUU is going to be phenylalanine. But how do you determine what nucleotides actually get used in your nucleotide sequence? So this brings us to the concept of the open reading frame. So an open reading frame is a stretch of nucleotides that can be made into protein. It doesn't necessarily get made into protein. But if you look at the different now reading frames, forget the open to start with here, we've got 3 different reading frames down here. It's a reading frame that starts at nucleotide 1, a reading frame that starts at nucleotide 2, and a reading frame that starts at nucleotide 3. Remember, it's a non-overlapping code. This is what was shown mathematically and then later in terms of experimentally. So here, if we start at nucleotide 1, we have AUG, which is what? That's the one I expect you to remember, methionine and start. And then the next codon, the next codon, next codon, next codon, next codon, next codon. And eventually gets to a stop codon. Every 3 nucleotides. However, if you start here at nucleotide 2, you've got a stop codon, no start codons, another stop codon, a bunch of other codons here, but no start. So reading frame 1 here is an open reading frame because it's got a start and a stop. The third reading frame down here at the bottom, where you start at nucleotide 3, could be an open reading frame because there may be nucleotides over here on this side, there may be some more nucleotides over here, but there are no stop codons that are in here. So it could be part of an open reading frame. And just by looking at your nucleotide sequence doesn't necessarily tell you what protein is actually going to be made there. There are 3 different possibilities from this one messenger RNA. And if you think about DNA, when we talked about transcription, either of those strands can be being transcribed. So one stretch of double-stranded DNA actually has 6 different reading frames in it. So how do you know which one's actually being used? You can guess that you've got an open reading frame that's got a start codon and a stop codon, but that's only a guess. And so you really actually have to go in and find out what the proteins are that are actually being made to know which of the reading frames are actually being used here. You can figure out what the transcripts are, and so that'll at least lower the possibility down to 3 potential reading frames that could actually be being used. So what interacts with each of these codons? It's our friend, the tRNA, originally actually known as soluble RNA, sRNA, to differentiate it from the ribosomal RNA, which was non-soluble. tRNAs have this pretty typical structure. Everybody draws them as clover leaves. That's not what it really looks like. It's more like an L structure, which we'll see on the next slide. But it's a little bit easier to look at this particular sequence here. So 5' to 3'. The 3' end has an OH. That's where the amino acid gets added. And we'll see how that happens next time. It's the amino-acyl tRNA synthetase that puts this on. Many tRNAs have CCA at this end. Sometimes those are added separately. But in some cases, they're actually coded for as well. If you thought that modification was happening a lot in ribosomal RNA, it's nothing compared to the modification that happens in tRNA. Usually about 10% of the nucleotides in tRNAs, and this is just one tRNA here, are modified. They'll either be methyl groups, either on the bases. You can also have methylation that happens on the ribose. You've got pseudouridine. That's what this psi here stands for. And we'll talk a lot more about this particular modification here, inosine and methylinosine. This particular part here is the anticodon. And again, what you need to remember is that we're going from 5' to 3', and so that means that nucleotide 1 of your anticodon is actually going to be base pairing with nucleotide 3 of your codon, because they're always going to be in opposite orientation. These guys form these base paired structures. Some of them are actually your non-canonical base pairs. When I say non-canonical, non-Watson-Crick. And so they're not always going to be AU and GC base pairs. I'm trying to see if there are any good examples on here. You've got GG, GC base pairs as well. So as I mentioned before, this is what the tRNA actually really looks like, because whenever you talk about RNA being base paired, it's going to be helically paired together. And this is color-coded the way that normally people will draw the cloverleaf structure. The D arm, because it's a dihydrouridine, which is always present here, and the T arm, because there's always a thymidine in this arm, basically wrapped together in the middle. You've got your 3' OH, which is where you're going to have the association with the amino acid, and then the anticodon loop being down here. So tRNAs coding for the individual amino acids, but we've still got 64 codons and only 20 amino acids. How is that dealt with? A certain fellow by the name of Crick, where's that name sound familiar from? Same Crick as Watson and Crick, came up with this idea that he called the Wobble hypothesis. And all that the Wobble hypothesis says is that the first nucleotide of your anticodon doesn't have to form a normal base pair with a third nucleotide of the codon. And that's just shown down here. Here we have your tRNA on the top, the messenger RNA on the bottom. Again, 5' to 3', if you're talking about the tRNA, so your first nucleotide in the anticodon may or may not form a normal, quote-unquote, Watson-Crick base pair with a third nucleotide of your codon. And so here, specifically, we've got a leucine tRNA that has a UAG, again going from 5' to 3', anticodon, that U can pair with A, but it can also pair with G. A different leucine tRNA, the first position of the anticodon G, can pair with U as well as pair with C, and then this one that I pointed out last time, inosine, which is a modified nucleotide, can pair with U, C, or A. And what that means is you can have one tRNA that can recognize more than one codon. So you don't actually have to have 61 different tRNAs, each of which has an anticodon that's exactly going to base pair with each of the 61 coding codons. 3, of course, are your stop codons. So this way you can get away with having some of those, those differences. So I want to talk briefly about some of those modifications. Certainly inosine is one of them. Turns out that there are lots of other ones. In eukaryotes, these tRNAs are mostly made by RNA polymerase III, and they're made as precursors, just like what happens with messenger RNA and just like what happens with ribosomal RNA. They get chopped, they get pieces that are chopped out of them, and it turns out a lot of the splicing that happens in tRNAs, and this is in fact an image of a tRNA which is being spliced. This purple portion here is cut out of the tRNA. Very often, these introns are actually in the anticodon. So this tRNA as it's made is not going to be able to work until this gets taken out. And it turns out that a lot of this splicing is also splicing which happens in the absence of other proteins. This particular one that we just looked at was in the presence of proteins. That is one of the processes, this whole self-splicing intron, which is the whole focus of Dr. Lehman's research up here in the chemistry department. There are also lots of modifications, as I mentioned before, that happen to ribosomal RNA, and that's the focus of Dr. Water-Royal's group, also here in the chemistry department. What do some of those modified nucleotides look like? Again, there are at least 50 different modifications, some really wacko and crazy, but this is a nice example here. Just dimethyl G, 2 methyl groups that get added here. Dihydrouridine, as we mentioned before. Sulfur instead of your oxygen. A G deamination, that's your inosine. It turns out that many of these modifications are absolutely critical. If you don't have these modified nucleotides, you end up with all kinds of problems, and in fact some of the nutritional supplements that you'll get in your breakfast cereal, if we still eat cereal, we have any time between our latte and our muffins, a lot of these are modified nucleotides. And so you can actually correct for some of the defects that people have in some of the enzymes that are important for making these just in dietary supplements. So we'll finish out talking about inosine. Again, this is basically deaminated adenosine. Why is it so important? Because it can base pair with adenosine, uridine, and cytosine. And so it's very often present at the 5' position in the anticodon. One of the reasons that it works so well is that it's only forming 2 hydrogen bonds, but can form 2 hydrogen bonds quite nicely with adenosine, just as well as with cytidine and with uridine. And because of that, it's very often present here at the 5' position of the anticodon. Again, it's the Wobble position, according to Francis Crick, and allows you to have fewer tRNAs than you actually have codons. So we'll stop here. We'll talk about amino acyl tRNA synthetases on Friday.